thank you all very much for joining us this afternoon, this morning, this evening, whatever time it is, wherever you are. And I, just before I start, I'd just like to emphasize that the chat is open. And so throughout this next hour, do put any questions or anything else you'd like answered in the chat, and then I'll try and bring them in at the appropriate time. So this, I think, is the 12th one of these that we've done. We started last year, and this will be the last one until probably 2024. And that is because, as Eli says, we've got the WPC meeting next year in Barcelona, so you can actually not need to join a webinar. You can actually meet the scientists face to face. So as you know, the point of this is to, as Eli says, to put a face to the scientist, especially young, emerging, brilliant scientists, of which Janelle is one such person. And it's to sort of try and bridge that gap between what people do in the lab and what it actually means for the condition itself. So how can these lab-based work actually impact on what we do in the clinic? And that's very much the WPC sort of mandate, if you like, is to try and bring all the interested stakeholders and partners together to better understand and then to treat and hopefully cure this condition. So we've been very fortunate with this series because we've had support from Supernus Pharmaceuticals and Kyowa Kirin, and they have funded today's Research Spotlight. So thank you very much, and thank you very much for your support during this series. So it's a great pleasure for me to introduce Janelle. And I'm never very good on Janelle's surname because I'm not very good at French, but I'm going to go Drouin-Ouellet. Was that pretty good?
It was pretty good, yeah.
Yeah, thank you very much. And that is a certain type of French, and Janelle comes from another type of French because Janelle is in the University of Montreal. So I thought it'd be useful, Janelle, just to tell us what you're doing at the moment, because I know you're actually in Montreal, but you're actually in San Diego.
Right. I'm currently at the neuroscience meeting in San Diego. So that's the biggest meeting annually for neuroscientists. I think we are 30,000 people here.
30,000 people. So a small village of people on all aspects of neuroscience. So you're at the University of Montreal, but if we go back to the beginning, I mean, when did you, it's always useful to find out how people ended up where they've ended up. I mean, I know you're in Montreal, and I think you began life in Quebec, which isn't that far away, but tell us about what you did as a sort of your degree and then your PhD and why you chose to do what you did.
Right. So as an undergrad, I went to do biological medicine. So that was essentially the only program that would allow me to become a medical researcher. And this is what I've wanted to do since I was 16, actually. So I went to do that. And then I knew I wanted to do my graduate studies. Didn't know in what field exactly, but I got really interested already then in Parkinson's disease because of cell-based therapy.
Okay, so you always wanted to do medical research. So you
yeah.
always, it wasn't as though you were looking for really basic stuff. You always wanted something that had an application to the clinic.
Right.
Yeah. And so then, and you were interested in cell therapies and you were interested in Parkinson's. That emerged as an undergraduate or when you were starting to do your PhD?
When I was looking for a summer internship to do research in a lab for a short stay. And that's when I...
So these are the sort of things you do a couple of months over the summer in between your university years. Yeah. Yeah,
exactly. And I ended up doing an internship in the lab where I did my graduate studies. I didn't work on cell transplantation for my graduate studies, but that was what got me into the field. Yeah.
And what did you do during your PhD? Whose lab did you do that in, and what did you actually?
So I did my PhD in Quebec City in the lab of Francesca Cicchetti.
Yeah.
And we were working, so my thesis was about the role of inflammation in Parkinson's disease. And I was working with mouse models at the time.
Okay. So the idea there was that you'd be trying to study Parkinson's disease in a mouse. How much did inflammation start or drive the disease process?
Exactly.
And was that successful?
To some extent, yes. So I think the work we were doing, so that was 15 years ago, maybe. The work we were doing is now becoming more relevant, I think.
Right.
Yeah.
And so after you finished your PhD, where did you go next? And what was the mission of that project?
So during my PhD, I sort of I wasn't really satisfied with the animal models that we had at the time. And I wanted to do something a bit more closer to the clinic. So this is why I moved to do a postdoc in your lab, Roger, in Cambridge.
I remember that. I wasn't
good.
amnesia, but I can't even remember that you were here.
So, and the plan was to develop those patient-derived models. So generate cells from accessible sources such as the skin and blood, and be able to obtain brain cells in a dish to study disease mechanisms.
And then I think you started that project here because it was a bit of a labor of love, because it's not been an easy project.
I think we were ambitious.
Yeah.
And we thought we would be successful in maybe 3 years, but actually it took us about 10 years, I would say, by now. So
from here, from in Cambridge here in the UK, where you started the project, you then moved, I think, to Lund in Sweden.
Correct. So I moved to Malin Palmer's lab. So I think Malin did a spotlight as well recently. And Malin was one of the pioneers of the technology that we do in my lab, which is direct conversion of skin cells to brain cells. So that's why. So with the patient samples that we got in your clinic, then I moved to Malin's lab to really work on developing the technology properly.
So basically, you did your sort of undergraduate, your PhD in Quebec. You came to Cambridge for a few years, then you went to Lund for a few years, and you've been in your current position in Montreal for 4 years. 4 years. Yeah. Okay. Running your own group there. Yeah. And so let's so you come back a few hundred miles from where you began, so this technology, just explain. So I think a lot of people here will be familiar with the idea that you take an adult cell, you reprogram it to a sort of induced pluripotent stem cell, and then you turn that into a nerve cell. And we've discussed that in terms of replacing dopamine cells using that technology, trying to model disease. But you do things slightly differently.
Yeah. So the reason why we didn't want to use those pluripotent stem cells is because I really wanted to focus on idiopathic Parkinson's. So this is the type of Parkinson's where we don't know the cause really. So it might be genetics, but it's not one mutation in one gene. And most of the field is focusing on familial Parkinson's, right, where you have mutations. So I wanted to really try to develop a model that would be helpful for the 90% that for which we don't know the cause. And so the main risk factor for idiopathic Parkinson's disease is aging. So it's age. So the problem or the drawback, I would say, with stem cells is that when you take a skin cell and you make a stem cell out of it, you rejuvenate the cell completely. So because Parkinson's is such a strong association for Parkinson with aging, then you lose completely a big component of it. So
if you took a skin cell and made it into an iPS cell, you've essentially turned it back into the youngest possible cell. So anything to do with aging has gone. Is that right?
Yeah. Most of things that has to do with aging, which is why when you study Parkinson's in those cells, oftentimes you don't see anything relevant to Parkinson's because they're too young, right? You don't develop Parkinson as a fetus. So that's why we thought, well, let's skip that stage, right? That stem cell stage. Let's just go from a fibroblast, so a skin cell, directly to a brain cell. And the hope was that we would maintain the age in the cell.
Okay. So the idea would be that you would get a bit of skin from someone with Parkinson's, not a known gene, so-called sporadic idiopathic disease. You'd grow up the skin cells, the fibroblasts, and then you would directly turn that into a brain nerve cell, a neuron in the dish. And by so doing, you would have got the cells from the patients who've declared themselves as having Parkinson's, and you've retained age in it.
Exactly.
And that has the advantage you've retained age and you've got it from a patient, so you know that they're relevant. But so that seems to me very logical, very obvious, but presumably, given you've just told me it took you many years, it's not that easy to do. So what's the problem with it? Why isn't everybody doing this?
Well, it took a while for us to be able to figure out how to make this efficient. So the problem at the beginning is that we were successful, but with mouse cells, so that was not very clinically relevant.
Yeah.
And then with fetal cells, but again, Parkinson is not a disease of the developmental stage. So when the skin is formed, when skin cells are differentiated and they're in their final state, they don't want to become something else. They don't want to become brain cells. So you have some reprogramming roadblocks that will prevent the conversion of one cell type to another. So we had to figure out what was preventing this conversion and remove it in the cell to make it efficient. So that took a while, but now we have a good method.
Okay. So, I mean, that sort of makes sense that if you're a skin cell, you'd want to stay as a skin cell. You wouldn't suddenly decide that one day you're going to wake up and be a brain cell. Otherwise, your body is a little bit unstable in that sense.
You would hope that wouldn't happen. So, yeah.
And so, so obviously you can work out the technology by which to sort of reprogram it, or I don't know what the term is, transdifferentiation. You differentiate it from one to another.
Sure.
I mean, how efficient is that process? So how easy is it to do it? I mean, you obviously said there was some problems in trying to understand what factors you needed to give in order to persuade it. So how many factors does it involve, and how efficient is it? So if I took 100 skin cells, would I get 100 nerve cells, or 200 nerve cells, or one nerve cell? What would be the...
Yes. So usually, with the first method that we developed, you convert one skin cell to one brain cell. So it's a 1:1 ratio. So pretty efficient, but the problem with that is that we don't really generate the dopamine cells that are degenerating Parkinson. So to make it a bit more relevant, we had to optimize the method to make dopamine cells, and that's when it became less efficient, because you needed to introduce more reprogramming factors into the cell, so that made it less efficient.
And how many reprogramming factors are we talking about here? So how many would you need in order to turn a skin cell into a dopamine nerve cell, roughly?
So we're using 8 different factors at the moment, which means that, and we infect them with viral vectors, essentially. So
do you put in 8 separate viral vectors, or do you use one viral vector that has 8 things in it?
8 separate. So that's why it's not as efficient as the vector we made. That's a single vector containing 4 reprogramming factors. Then it's really efficient, because you can infect the cells and it will convert to a neuron.
Okay, and those nerve cells which you get retain age, and we'll come back to them in a moment, because we've got a question about what is aging and what have we retained with aging. So you can convert them, and how mature are those cells? Because obviously, you convert the skin cell, the fibroblast, into a neuron, into a nerve cell. I mean, how mature is that? Is that because it's retained age, but is it an old nerve cell, or is it an old nerve cell that's still
I would say it's an old cell, but it's not an old nerve cell.
Okay.
In the sense that there is a maturation process, because we completely convert something into a new neuron, right?
Yeah.
So you need to wait a few months for the cell to be fully mature. But in terms of cellular aging, everything is there, because all those signs were in the fibroblast, the parental skin cell. So it stayed in the cell. So the cell is old, but the neuron is not mature, if that makes sense.
Yeah. So basically, it's a new nerve cell, but it's an old new nerve cell, and if you left it for a period of time, it would age and mature.
Yes.
Yeah. Whereas I guess with the IPS, the reprogrammed, you can obviously convert them into young nerve cells, and then you can keep them for a long time in a dish, and they mature, but they're essentially still very young nerve cells. So what are the peculiarities of aging? How would you define aging? I mean, why is aging critical for getting Parkinson's? I mean, why is aging? I mean, we talk about aging and Parkinson's, but obviously aging brings with it a whole host of, as I know only too well, all sorts of issues, medical issues. But in this particular issue, what is it specifically about aging that's important for Parkinson's, do you think?
That's such an interesting question, and that's one I'm trying to answer, actually, because when we talk specifically about neuronal aging, so aging of a brain cell or the nerve cell, we don't really have model systems to study this. So one particularity of a neuron is that we're born with a set of neurons, and they will progressively die, but we don't get new neurons that are generated. For most of the brain. So that means that the neuron that is formed when we are born is going to live for 80, 85 years old, depending on your age. And we don't have a system in which we can study 80-year-old neurons, right? So most of the studies are based on rodents, which have a 2-year lifespan. So that's probably not completely the same as an 80-year-old neuron. So I don't think we really know. And when you talk about cellular aging, the first thing that is mentioned is usually senescence. So it's a state of a cell where the cell that usually divides will stop dividing, will maybe adopt a more toxic state, a bit like secreting toxic factors and stuff like that. But for a neuron that doesn't divide to begin with, what does that mean? Is there senescence in a neuron? We don't really know. I mean, I think there's not enough information. We need new models to study that.
But when you say I've got an aging nerve cell, how do you know it's aging then? If you don't,
right? So there are some hallmarks of aging, such as so telomere attrition. I don't know if everyone is familiar.
I'm not sure. Telomeres, are they something to do with controlling DNA or something?
Yeah, so it controls DNA, and so these are like on chromosomes. And they get shorter as you age. And at some point, there's some sort of a what's the word? I'm blanking on the word, but
they can't get any shorter.
Yes, correct.
They reach a sort of limit.
Yeah, and the cell will start functioning badly, let's say. Then there is usually oxidative stress is associated with aging. Gene expression also will change. Something that we call epigenetics, which is sort of, it's not new, but I mean, I don't know if everyone is familiar with it, but as you age, you will get genes that will be suppressed. So they will not being expressed because of something that will stick to the DNA and prevent their expression.
So this sort of epigenetics, so it's called epigenetics because it's sort of next door to the genetics, and it's this sort of this methylation. I think people talk about DNA and things like that. So aging, you could look at telomere length, which would tell you something about aging. You could look at the sort of epigenetic profiling, and telomere lengths would then influence when you can repair DNA. Is that what it's about, or is it? I mean, why does telomere length shorten and then make the cell unwell? Is it?
I mean, I'm not sure actually. I mean,
I know it's a hallmark, and I don't know the answer either. I mean, epigenetics, I can see that you could end up expressing genes which you wouldn't normally. So so marks of aging, I guess, are telomere length, epigenetics, as you say, sort of oxidative stress. How cells produce stressful would be another factor. And so, do you use all of this to sort of measure that you've got an old cell? So is that how you know that your induced neurons, which you've made from skin cells, are in fact aging?
I mean, we focused on a hallmark that we thought would be relevant to Parkinson's. So we know, for instance, that in the nerve cells that we generate, we have a lot of DNA break. So as you age, sometimes your skin cells will acquire a mutation. Your DNA will break because of, I don't know, sun rays or something like that will induce a mutation in your DNA. And usually the cell is pretty good at repairing it. And it will be like nothing ever happened. But as you age, this system is becoming less efficient. So those mutations are not getting repaired. And this is why, some people can develop cancer and that sort of thing, but also it can induce other things that pertains to neurological disorders. So we maintain this increase in DNA mutations in those cells. So that's another interesting thing, I think, associated with the model.
So your model is you make these induced neurons, which have the hallmarks of aging, And then presumably, so we've talked about how you characterize it, and I think there's things in the nuclear envelope and things like that. So there are different markers you can use.
Yeah.
Then one of the questions is about maturation. So we've said the cells are immature. So how long do you leave them in the dish for them to mature? I mean, can you leave them for like a year, or how long do you leave them till you think you've made them into mature dopamine nerve cells?
Right. So they are difficult to maintain for long term. So maybe not a year. We've maintained them for 4 months.
Okay.
And they were
so the experiment would be get some skin cells from a patient,
yeah,
transdifferentiate them into nerve cells, which takes what, a couple of weeks?
25 days.
25 days. And then you grow them for another sort of 4 months, you said? Did you say, or 4 weeks?
Yeah, so that's the longest experiment we've done, just to see, to push it.
And at the end of that, how mature are those cells? Would they look like the normal dopamine cells you'd expect to find in the adult human?
No, because one of the main characteristic of dopamine cells is that they have these big projections, big neurites. And in a dish, we don't really have that. But in a dish, we don't really replicate the brain environment, right? So we have many other cell types that are really important to nerve cells in the brain, and we don't currently have that in our system. So of course, it's why it's more difficult to maintain a healthy neuron for long term. So when we look at Parkinson's disease associated signs in those cells, we usually do it within a month, because we already see those signs at that stage of the neuron.
So basically, grow the cells. 25 days later, I've got some nerve cells made from a patient's skin fibroblasts. I then leave them for a few weeks till they start to mature. As you say, it's a very sort of reductionist model. You're sort of saying the only thing I'm going to be studying here are the nerve cells from the patient's skin cells. So if there's inflammation, which you were talking about as part of the work you did earlier in your career, if I think the glial cells, these astrocytes, microglia that we've heard at before might be important, we're not really talking about those. So we're only looking about intrinsic problems in the nerve cells. So one of the questions is about in your model.
I was just gonna say that we are working to generate, so we can make astrocytes now, and we are trying to make the resident immune cell of the brain as well to study
using the same technology. So you take a skin cell, turn it directly into a nerve cell, directly into an astrocyte, directly into these other cells, and then mix
so that we would generate them with the signs of cellular aging as well.
Okay, well, I'll come back to that in a minute because I'm quite interested in that. And so when you're trying to compare your patient cells to see if there's a problem, what do you use as your comparator? Do you take skin cells from people without Parkinson's disease, or do you just sort of say, well, actually, I know roughly what they should do, so I'll just see what I see that's wrong here?
No, so the two main risk factors for Parkinson's are age and sex.
Yeah.
So this is why it's very important that we compare the neurons derived from the patient to age and sex match controls.
Okay, so your experiment is, so we're getting down to the nitty-gritty because I want to know what you found. So you've got your skin cells from a group of people with Parkinson's, and presumably the advantage of this technique is, given you're just taking one patient skin cells turning into nerve cells, you could do dozens of patients, whereas I know if you're making induced pluripotent stem cells, you take one patient, you have to reprogram them, you have to choose the clone you want, you grow it up, you differentiate it. Very expensive, very labor intensive. Presumably with this, you could do a lot of patients quite quickly.
That's correct. So the first study that we just did, that was published recently, we had 30 lines, I think. So
30 different patients.
Including, 18 patients and 10 controls, so 28 lines.
Okay.
Which is much bigger, much larger than usually what you would see with iPSC studies.
Okay. So the experiment is we get say a couple of dozen people with Parkinson's, get a couple of dozen people age and sex matched, take their skin cells, turn them into nerve cells, dopamine nerve cells, let them mature, and we've shown that they retain the age factors. And then what do you find? So what's different between those? Is there any big difference between what you see in the Parkinsonian nerve cells, if you like?
Yeah, so we first focus our investigation on a cell process that we knew would be present in neurons, right? Because of the fact that we don't have other components of the brain. So we focus on protein degradation system.
Okay.
So the neuron needs to be able to recycle
Yeah.
damaged proteins and stuff
Okay.
like that. And as you age, this becomes less efficient.
Okay.
So which brings toxicity to the cell.
Okay.
So we looked at that in those neurons that we would generate from the skin cells. And we saw actually big differences between the cells derived from the patient and the matched controls. So we would see accumulation of those proteins, so a decrease in efficiency of removal of those damaged proteins. And this is the same that we're seeing with mitochondria, which is the powerhouse of the cell, very important in Parkinson's, because it becomes dysfunctional in a lot of familial forms.
Okay.
So we think it's a good target for Parkinson's. And we see that they're not getting properly removed when damaged in the patient lines. Yeah.
And when you looked at them, did they look different? So if you gave me a dish and said, look, here's a bunch, Roger, here's a bunch of nerve cells, can you, which I've grown from patient skin cells, can you see a difference? Could you see any obvious differences or?
Not obvious. We did some analysis where we saw, there was one experiment that I did at first, and I saw that the cell body of the patient-derived lines were bigger. And I did not investigate that further, but that was a striking difference.
Yeah.
And, I'm not sure what it means, but
okay,
it was different. Yeah.
So the differences you're seeing, so looking down the sort of microscope at the cells, you say there's not an obvious difference. So it's not as I look in and say all the Parkinson's nerve cells have died and the control ones. They look pretty similar, but there's obviously relatively subtle differences. So one of them is that the problem you have is that the ability to get rid of proteins and therefore maintain the health of the cell is compromised to some extent. And the mitochondria, the ability to generate the energy which the cell needs in order to survive, is also compromised. And was that was seen across all of your Parkinson patients, or?
No. So probably for both, protein removal and mitochondria removal, we would see it in about half the lines. Okay. And that's what's, I think, great about the model, is that because we can work with many, many lines, we can actually start to dissect out like who has impairment, who hasn't, and can we figure out why this subgroup has mitochondria problems and this subgroup doesn't, right? Because Parkinson's is so heterogeneous in terms of clinical presentation. I mean, I think it's not completely out there to think that maybe there's something different in the cells as well, right? Reflecting these differences in the clinic. So that's what we're trying to figure out with the model, because we can work with large cohorts of patients and try to figure out at the molecular level what are the differences.
So ultimately, you could say that anyone coming to clinic, in theory, and I'm not saying this is going to happen because obviously there's a cost implication and a labor, but in theory, anyone coming to clinic could have a skin biopsy because they're relatively innocuous little, and then you grow up the cells, turn them into nerve cells. We give them to Janelle and say, tell me where the problem lies there. And then we could go back to the clinic and say, your problem seems to be in protein degradation, whereas the next patient, your problem seems to be in mitochondria. The next patient may be in this thing called autophagy, getting rid of proteins through lysosomes or something. So you can start to work out what's wrong at the individual level.
Yeah, so that's the hope in the very long term, but in the short, medium term, I would say, I would like to be able to use this for patient selection refinement in clinical trials, because if you have a drug that targets mitochondria, and that, I don't know, half the patients don't really have, it's the mitochondrial problems is not driving the problems, then you would only select those for which they would benefit directly from having their mitochondria saved, let's say.
So the way forward would be, based on what you just said, then if I've understood it, is, you sort of say, well, I've got, 100 lines in growing. These 25 patients seem to have some problem with mitochondria. They would be the ones I'd target for the therapy.
Correct.
And have you done any work where you've, so there's a question here about sex differences, whether you've seen any difference between male and female cells, and there was another one about age. And I suppose one which would be thrown in there is genetic forms of Parkinson's disease, which, as you said, a very small percentage of actually people with Parkinson's disease have a known single gene causing it. So how much have you looked at sex differences? How much have you looked at younger onset versus older onset, which of course is the advantage of your system? And how much have you looked at any particular genetic types?
Right. So great questions. So about sex, in what we've looked at in nerve cells, we haven't seen striking differences. And based on the literature, I expect that the difference will be driven by immune cells. Okay. And other factors such as hormones.
So when you say the difference between male and females, yes. So any sex differences will be driven not by a problem inside nerve cells, but by other cells interacting on it. So the
hormonal and immune system.
Okay.
And we actually have a project in the lab looking at that, but in the brain resident immune cells. Yeah.
Okay. Yeah.
And so about the
that's based on the premise that Parkinson's is a bit more common in men than women, and therefore, and I think also there's some data, because we've just been looking at clinical progression, that the disease progresses slightly different in males and females. So,
interesting.
Yeah. Okay.
Yeah. So we're trying to figure out the mechanisms behind this, of what's happening in the body of a man and a woman. Why is it different? And in terms of genetics, so in any type of analysis that we're doing, we have some genetic PD lines, but they're in very low numbers, but we always include them to compare, right, with the idiopathic PD patients. But we haven't really seen thus far anything strikingly different, I would say.
And which genetic forms have you seen of these?
So we have Parkin mutations, and we have
the very young onset form of it.
Yeah, and LARC2, which is for which age is important as well, very much so.
Well, LARC2 is rather complicated because some people get Parkinson, some people get Parkinson's dementia, and some people get nothing. So
right. Exactly.
Okay. So you've looked at some genetics, but you'd say I can't really answer that question because I haven't got enough lines to be able to.
Correct.
But I suppose what you'd say is there's no striking difference.
Not with the 2 lines we have per gene. Yeah.
Okay. So you haven't seen any sex differences of note. You haven't seen any genetics. What about age? Have you looked at young Parkinson's versus old Parkinson's using this technology? Yeah.
So when we try to, let's say, subgroup patients based on our findings, age is usually what so the signs are more severe in older people. Age at onset would be the second most important factor thus far in what we find. So older age at onset is associated with more problems in protein degradation.
Okay. So it's older. So the older you go, the more obvious the deficits appear. Yeah. Okay. And have you been able to correlate anything you see in your cells with other aspects of Parkinson's? So can you sort of say, well, actually, it's not just that I've seen a problem in protein degradation or mitochondria, but actually when it's the more pronounced that is, the faster has been the clinical course in that patient?
Not really, no. There was, we saw an association between the accumulation of pathological alpha-synuclein and the rate of motor decline, but we have too low numbers to really conclude anything about this, but there were signs of it, but I can't say that, this
so
is
that's the accumulation of the abnormal protein associated with Parkinson's was greater in cells in people who seem to progress more quickly in terms of their motor disability and such like. Okay. And so going forward, you've obviously, there are a number of lines in which you could take this, if you don't mind the pun. So one is obviously you can use it to select or enrich a population for your trial. What about drug screening? So obviously people are very keen on the fact you just grow lots of cells, you chuck them, 4,000 compounds on them, and can I reverse? So you can imagine in your experiments, I take the cells which are abnormal, I go to some drug library or compound library, I put all the compounds on, can I reverse them? And if I can I therefore reverse translate and take it to the clinic to treat people? Yeah,
so I see this combined with personalized medicine So if we manage to reach that stage where you go to the clinic, get your diagnosis, pee in a cup, because we're trying to also make nerve cells out of cells collected in urine to make it less invasive. But let's say for now, you give a piece of skin, small piece of skin, and then we make neurons and do this drug screening, and that would inform how to best tailor the therapy for your Parkinson's disease. So I mean, that would be another way. So right now we're trying to subgroup the types of Parkinson's disease to refine patient selection in trials, but that would be perhaps another way in which we could use this technology.
Okay. So, and do you think going forward that this is something we should be building into our trials? Do you think everybody who comes into a clinical trial, we should at least take a bit of skin from them, even if we don't have the resources to turn them into nerve cells, and just bank the fibroblasts, so that if we see something in a subgroup, we could use your technology to then go back and interrogate that?
Yeah, I mean, that would be, well, if patients want to give skin, we're always we'll take it, because we need to build up numbers. We need to study a large cohort of patients to be able to do what we plan to do. We need to really increase the number. So of course, but I don't know if freezing, so that's interesting. And that's why I really want to be able to make neurons out of urine samples, because what we want to do as well is follow-up studies where people with signs of, prodromal Parkinson, so sleep disorders, or maybe loss of sense of smell, and that kind of indicators that are not 100%, but that could mean that you are developing slowly PD.
Yeah, well, we had Ron Postuma on here.
All right.
And he's right next door to you, isn't he?
Yeah, he's a different university, but yeah. So the goal would be to take urine samples every, I don't know, couple of years to follow up these people and see if we can figure out what in the nerve cells that we generate, are there any changes, to be able to single out people that will go on to develop PD in 5 years, because then we could maybe intervene earlier before the damage is done.
Okay. And how efficient is that? So this urine, I mean, how many cells do you get out of not
that many. So that between 2,000 and 7,000 of a pretty good sample size.
And what are they? They're obviously things that come out of the collecting tubules of the
renal epithelium, the bladder.
And do they reprogram as easily into nerve cells as, say, the skin?
Not yet, but we'll get there. But we were successful in reprogramming. So the proof of concept, we did it, but now we need to optimize it. Yeah.
And do you have any evidence for what you were just saying, which is very exciting, that if you collect cells sequentially over time, that the nerve cells you generate are different, taking outside age, because obviously you're going to get older. But if I was to take someone with Pugsies and take where skin, blood, urine, grow them at time 0, 2 years, 4 years, 6 years later, do you have any evidence that they change over that period of time, or is this merely something that you're hypothesizing at the moment?
This is completely theoretical. We need to try it, right, and see. But those experiments are, the long term, they're expensive. So this is not something that we've started yet, but it's something I'm planning to do eventually.
So going forward, we can use this technology to select patients for trials. We may want to prospectively put it into trials, so we've got samples to go back and look at people. You're obviously trying to develop more convenient ways to do it. You pee into a pot and that's sent off and it's all dealt with. So just tell us about these other works you're doing about, because a couple of other things I want to explore was about making other cells. So obviously, as you say, how can you more faithfully use your aging neuronal system to better model Pugsies? Because we've already talked about the fact that we don't have any other cells. So just tell us how you're making the other cells and how you see that working out. Would it be that you make different cells, put them in a dish, and then add the patient's blood to it to look at the immune cells? Or how do you see this evolving?
Yeah, so for now, the goal is so I'm interested in 3 cell types that are in the brain. So nerve cells we have already. We can make glia, the astrocytes, which more and more people are getting interested in those in terms of how this drives Parkinson's. and microglia, which is the resident immune cell. So the goal would be, so now we can make two of them, the nerve and the astroglia. And the goal would be to do a multicellular system, so 3D tissue, sort of
a little sort of organ, something. So it's this three-dimensional bit of midbrain, the area that normally contains dopamine cells, and you would have your induced dopamine cells, your induced astroglia, and your induced microglia.
Yeah, so all of these would maintain hopefully the age of the donor, so we could study the interaction between the different cells. And one cool thing that we can do is to reprogram those same skin cells into pluripotent stem cells to rejuvenate them. And then we could add, let's say, young astrocytes to the mix, right? And see how that's different than when you have old astrocytes and how that influences neurons.
I see. So that would be a way in which you could dissociate age from patient specific, because they come from the same patient, and the only difference between them would be one's an old nerve cell or an old astrocyte.
Yeah, because what we see currently, what we did that, so we rejuvenated some of the patient lines, and we would wipe out completely the Parkinson pathology.
Okay, so all these problems you were saying about protein clearance mitochondria, if I took those skin cells, rejuvenated them, and turned them into the dopamine cells, I wouldn't see anything wrong, as you've seen.
Yeah.
So that implies that your aging system, well, I don't know if it's more faithfully representing the disease. It definitely has problems with it, which the other cells don't. Okay. And when you're trying to reprogram into astrocytes, just as the way you were saying, when you reprogram into nerve cells, you're trying to reprogram into dopamine nerve cells, dopamine neurons of the midbrain, because they're the type lost in Parkinson's disease. Do you have to do the same with astrocytes? Do you have to reprogram them into the astrocytes of the midbrain, or are all astrocytes pretty much the same? It doesn't matter where you get them from.
No, they're different. They are different. And ideally, we would generate those that are in the midbrain. We're not there yet. We're just happy to see an astrocyte at the moment. So this is very, this is a young field. We're starting with this, but that would be a perfect scenario.
Because one of the earlier questions we had was about, making new nerve cells in the brain. And although it's quite controversial, I would say, I think most people would probably say that, I mean, new nerve cells are made in the adult human brain. I think in the subventricular zone, by the ventricles in the brain, going to the front of the brain for smell, as well as in the hippocampus for memory. I think the question has been the extent to which that really does happen in the adult to any significant extent. But I guess one of the questions there is, why does that happen there and doesn't happen in the midbrain? I mean,
that's an excellent question. I think there's not even enough evidence that this happens in human, and we don't know. And if it does, we don't know what the purpose of it. I think it's pretty clear that it's not exactly the same as in rodent, and most studies have been done in rodent for this type of newborn neurons.
I know it's very controversial, because the people who say we've definitely seen it, and then other people have said we can't see any new nerve cells in this part of the brain. So I think it's unclear. But I know one of the theories that was put forward was the astrocytes. The astrocytes, the hippocampus are encouraging new nerve cells, whereas in the midbrain they're not. So I guess one of the challenges for you will be the ability to make relevant cells for the disease.
But I think, if you look at the field of stem, making nerve cells and glial cells out of stem cells, it took probably 20 years to be where we are now, and we can actually generate those astrocytes that are region specific. So, and as technology gets better, we can do these sorts of things quicker. So I'm expecting that. So we're not there yet, but I think we will get there. It's a matter of time.
So your sort of vision is that ultimately you'll be able to make these little sort of three-dimensional structures of a patient's midbrain, recapitulating what goes wrong inside their head in their disease state.
Really, so I'm really interested in why everybody ages, right? But why some people develop Parkinson's as an effect of aging? And why some people just get older without Parkinson's?
And presumably it could be used for any condition in some ways. Given a lot of diseases age related, as we've already said, like Alzheimer's disease and other forms of sort of Parkinson's Parkinsonism could be to that. So one of the questions I have is about, could you exploit this technology? Because you talked a bit about cell therapies, and obviously we had Marlon and Agneta on talking about dopamine cell therapies from stem cell sources, but could you actually use your technology to reprogram cells in the brain? So obviously you're taking adult skin cells, turning them into dopamine cells. Could we not use a similar technology to say, can't we just inject whatever it is you put in your dish directly into the brain? Not because we have skin cells obviously in our brain, but we have astrocytes or other cells. Couldn't we just transdifferentiate a bunch of glial cells, which we don't really need, into dopamine cells in the brain and actually repair the brain? Or do you think we would just end up with the same problem, that we're making faulty dopamine cells and they will all fail?
Well, that's interesting. And this is funny because we had multiple debates when I was in Marlon's lab about this, because so this is something that that people are trying to do. And because we see so many signs of Parkinson's disease in those cells, that wouldn't be my approach. But I think that if we can figure out, so especially with in vivo reprogramming, where you would take a cell type that is toxic, and then reprogram it to replenish the normal population, I think that would be a brilliant approach. But I think we need to figure out how to erase the pathology in those cells that we want to convert. Because I think that if you make a nerve cell out of something that is old and toxic, you will create a neuron that is old and toxic. So, but that's, that's completely hypothetical, right?
So the worry would be that if we exploit your technology and, for example, say, well, Janelle's got this terrific way of making dopamine cells, I will inject those factors into the patient's brain and convert 250,000 astrocytes and dopamine cells. You're actually generating 250,000 dopamine cells that are unhealthy, because that's why you're doing it in the lab. But you don't want to do that if you're trying to repair the brain.
I think you would need to maybe modulate some genes that are driving aging and try to maybe repress that to rejuvenate to some instance the cell or something like that. I don't know what it is right now because we don't know, but we would need to have this kind of approach to make it viable for patients.
Okay, so going forward, obviously that as a technology probably is a different area, but I can see that making more lines from patients in trials, or using it as a way of selecting for trials, making more complex systems. What about moving outside? So obviously we always think of Parkinson's in terms of the dopamine cell loss, and there is no doubt there is dopamine cell loss, but obviously the cell loss isn't restricted to that site. So how easy would it be to use your approach to look at aging cells, which, these cholinergic nerve cells which sit at the base of the brain, project up to the cortex, the outside of the brain, heavily involved with thinking, might underlie some of the problems with the dementia and neuropsychiatric problems you see. How easy would it be to use your system, not just to make a complex midbrain, but to rebuild different bits of the brain or study different cell populations?
Right. So at the moment, we can make many different subtypes of nerve cells in the brain, including cholinergic.
Using your approach?
No, using other people's approach.
So making iPS cells and turning them?
No, directly converted.
Oh, directly converting. Okay, yeah.
I'm not aware of anyone who's done a mix of those cells to see how they interact. And so, it theoretically it's possible. We know how to make those, but no one has.
Do you think that would be a useful way forward? So, patient comes, I'm trying to think of the future and how, trying to think how we could really better improve our understanding and therefore treatment of patients in a personalized way, as you say. But we could take some skin cells, we turn them into dopamine cells, we turn them into cholinergic forebrain cells, we perhaps turn them into cortical nerve cells, we even perhaps turn them into enteric nerve cells, cells of the gut. And we say, well, we've got one patient, we've got 4 different types of cells. We could build these complex systems. We could look at all of these and we can see whether the pathology is similar, different, and such like. Do you think this is science fiction, or do you think this could happen?
No, I'm fairly confident that this could happen, because usually so this is happening to some extent right now with stem cells. So we call these assembloids. So you assemble, you make a gut in a dish, quote unquote gut in a dish. You make a mini brain in the dish, a midbrain, and then a forebrain, and you assemble them together. So you sort of create a human system in which you can have some pieces of synthetic organs, and you can study how they interact. And I think that there's no reason why we couldn't do that once we're good at making those cells. We can just assemble them, because now we culture them in a 3D system. So we can do organoids, let's say, with those directly converted cells. So it's a matter of assembling them. So I see that.
You can imagine that a patient would end up, so Janelle turns up, we take a bit of skin, and we then end up with a dish where we've got multiple different types of nerve cell, all growing in the same culture system, all talking to each other. And you could then study the pathology in each one, and presumably you could then perturb the system. You could inject some pathological synuclein into one site and see how it spreads and the pathology it causes. Presumably you can then try drugs across all of the cell populations and see, does it work better for some than the others?
Yeah, that would be pretty cool to do. Yeah.
Okay.
Not going to happen in the next few years, but that would be pretty cool.
Because I know people are interested in this sort of organ on a chip, you know, for drug screening, they sometimes take stem cells and they grow bits of liver, heart and such like, and then they test for toxicity of drugs. So this is very interesting that you could make more complicated, multiple different nerve cells from the same patient on a dish by which to study them all at the same time. And so if I, you know, in the last few minutes, I mean, if you had to see, you know, in the next 5 years where your work will go, and for any of the people on the call, you know, who have Parkinson's or know people with Parkinson's, where do you think the biggest impact will come in your work? Where do you see it really becoming a sort of major sort of, you know, impact?
Right. So I don't know if 5 years is feasible, but that would be for the patient selection refinement. So I think that if we keep increasing the amount of patient line we're working with, and try to see what is the dominance in pathology in those different patients, if we manage to subgroup them based on what we see, that would be the first application I see. Yeah.
Okay, and we've got a question here about rejuvenation, which of course I'm very keen on. You don't need to worry about it for a while, but I mean the idea that you could rejuvenate cells. So there's been these quite interesting experiments where people have used young serum, for example. So these parabiotic experiments where you take mice and you obviously can't do this in people, but in mice you take a young mouse and an old mouse and you join their circulation together, so the young blood goes into the old mouse and the old blood goes into the young mouse. And there's some evidence that the young blood and the serum in that makes the brain a bit younger. So do we know anything about how we could use your system to explore whether we could rejuvenate cells? Because obviously we're looking at better ways to treat Parkinson's and we're trying to look what the deficits are and treat the deficits. But obviously one way would be to erase age. I mean, obviously, if you knew the answer to this, you wouldn't be sitting there. You'd be in your island somewhere in the Bahamas with the money you've made from it. But I mean, do we know whether there's anything you can do to reverse aging outside of reprogramming?
Yeah, excellent question. And those studies are fascinating. And there's this, well, I don't know if it's a new field, but it's new to me anyway, this field about rejuvenation as a therapy.
Yeah.
Which I guess is controversial because that suggests that aging is a disease, but people are really trying to figure out how to make us live longer, essentially. Yeah. And the problem is that if we live longer, there will be more Parkinson's disease cases and that sort of thing. So that's why we need to figure out how aging and Parkinson's interact. But I don't know. So I don't think we've put our fingers on what factor is a rejuvenating factor other than those Yamanaka factor, those that can convert cells to stem cells. Yeah.
So reconverting cells. So yeah. So we've got no clear factors that rejuvenate things, and often
perhaps,
clinical are clearly
not my field. So I couldn't be able to. I can tell you.
Yeah, and I think there was a point of clarification here because someone's written about the older you are, the worse the Parkinson's disease is. I don't think we were saying that. I think the point you were making was that the older your cells, the more obvious are the deficits in it. But obviously, that's almost irrespective. So if you took control lines, so if you took people without Parkinson's and you looked at cells from people who are 90, 60, 30, you would see that at 90 they're not as good as they were at 60 as 30. But that doesn't.
Well, yes, but also we do see more protein degradation impairment in later age at onset.
Yeah. But we don't know, but I would just say, I suppose it doesn't mean that that disease will progress more quickly in that person. It just tells you that the deficit is more obvious in that. And presumably that's just making the point that the older you are, the more obvious are the deficits that you see when Parkinson's comes along. So I think it's important not to go away thinking if I get Parkinson's at 80, I'm going to do much worse than if I got it at 60 or 40. I mean, there are some age factors, but that's
we can't tell anything about that looking at the cells we make.
Yeah. Okay. So I mean, we're coming towards the end here. I think we've dealt with most of the questions that have come up on what people are asking. I mean, someone's asked about transplantation using young cells. I would say that transplantation is done with young cells because we use embryonic stem cells or iPS cells, which are reprogrammed, which, as you said at the beginning, are the young cells. So, I mean, all it leaves me to do is to say thank you very much, Janelle. I mean, this has been an amazing journey in terms of, you know, discovering how to do this. I mean, I don't think people should underestimate the problems with it because, of course, nature designed it that these cells, as you said at the beginning, once you're in one cell type, you will sell in one cell type. Why on earth would you want to change? And obviously, that's not good for you. So trying to work out how to do that has been a great labor of love. And, you know, many congratulations on getting that to work. And the fact that it's also, you know, having got it to work, get it to work efficiently. And your vision, I think, is very inspiring for many of the people here about how we could start to see personalized medicine in a real way. Because I think a lot of people talk about personalized medicine, but they don't really know what that means. So thank you very much. Thank you very much for this, you know, all the work you've done. We wish you all the best.
Thank you so much for having me.
No, it's been great having you. It's just, I find these sort of things so inspiring. I would like to thank everybody who's joined us this afternoon and people who are listening to the recording of this. As you know, we're not doing these research spotlights next year. And the reason we're not doing them next year is that we have the actual World Parkinson's Congress, which will be in Barcelona next year in July. If you want to, it's a great opportunity to meet not only researchers, but anyone who's got anything to do with Parkinson's, so clinicians, scientists, patients, carers, therapists, anything, anybody who's got anything to do with it will be there. They're normally there in hundreds and thousands. If you want to submit an abstract, then do so. The deadline for that is January the 13th. Our researchers put up regular blogs. That's how we tend to choose the people for the research spotlight. And they will carry on next year with regular science updates. So all of that, if you notice in the chat, is appearing there from Eli. So thank you all very much for joining this afternoon. I wish you a very happy Thanksgiving for those in America, Christmas and New Year, and look forward to seeing you all in Barcelona next July. And thank you again, Janelle, for a very, very inspiring hour of discussion. Thank you.
Thank you so much.